In this video, we are going to look at enzymes in biochemistry and how they undertake biochemical reaction in a biological system. This is the next tutorial, so don't forget to like, subscribe, and also share to whom it may concern after watching. And also press the notification bell so you can receive every bit of updates from this channel. So let's begin. Define what an enzyme is as they apply in biochemistry. So what then are enzymes? What are enzymes? So when we say enzymes, we say they are catalysts, as in they are biological catalysts because they are found in a human system and also other biological systems. So as a catalyst, they speed up the rate of a chemical reaction. So when we say a reaction rate, it just defines how fast or slow a reaction can go. So enzymes are just catalysts that are in a biological system that speed up the rate of a chemical reaction. So as said here, they increase the rate of a chemical reaction without themselves being changed in the process. So as enzymes act on the reaction to speed up the reaction rate, they don't get destroyed at the end of the reaction, or they are recovered so they can speed up another similar reaction. They are found in a human system and also other biological systems. They catalyze most of the biochemical reactions so that the reactions can occur very fast. So they don't change the reaction, but they can just let the reaction happen at a faster rate, or you can just let a reaction happen within a shortest possible time. That is how enzymes function. So let's take this as an enzyme activity. So what they do is they are proteins. So enzymes are proteins. They increase the rate of a chemical reaction by what we call lowering the activation energy, which we are going to look at in the next few slides. So let's say this is a reaction by carbon dioxide and water to produce a carbonic acid. So the enzyme that catalyzes reaction is called the carbonic anhydrase. If this reaction occurs without any enzyme, then this reaction will go through the whole of this pathway. So the one shown here, that is the one shown in pink, before it can get to carbonic acid. So the enzyme for this reaction is called carbonic anhydrase. Now when we add the enzyme to it, it will just go through this path. So within the shortest possible time, we are going to form our product, this carbonic acid. So if we have to go through this long path, it's going to take a long period of time before we can get to the carbonic acid. That is just the reaction without the enzyme. That's why we call it what? An uncatalyzed reaction. The uncatalyzed reaction. The EA here stands for the activation energy. The activation energy, which we are going to look at in the next few slides. So as shown here, as biological catalysts, they speed up almost all chemical reaction that occurs in a body. So they have 3D structure that fit their specific substrate. So that's how enzymes process their chemical reactions, or the chemical reactions that they catalyze in a biological system. So now let's look at how enzymes, or properties of enzymes. So as a catalyst, they say they have a higher catalytic efficiency. So when we hear of catalytic efficiency, it means that they are able to speed up the rate of chemical reaction at million folds or thousand folds. So if an enzyme, or if a reaction occurs at, let's say, the rate of X, and you add an enzyme, it is going to increase the rate by 10 to power 3 times, that's 1000 times to 10 to power 17 times. So the enzyme just speed up the rate of a chemical reaction by increasing it to a rate of a fold of about 10 to power 3 to 10 to power 17, faster than if it is not catalyzed. Enzymes are highly specific. That is to say, each enzyme in the system has a specific substrate they catalyze. So we can't have, let's say, two different enzymes going for different substrates. So every enzyme has just one and only one substrate. That defines their specificity. So they go for just one specific substrate. That is, enzymes are very specific, and that they catalyze a specific chemical reaction by converting a specific substrate to a product. Now enzymes are proteins, so they require the mild conditions in the body. They work best in the ambient temperatures, good physiological pH, and also ambient atmospheric pressure. So at a temperature of 37 degrees Celsius, most enzymes are active, and at the pH of around 7.0, most enzymes are active, and at good pressure, most enzymes are active. So these conditions are what we call the optimum conditions that enzymes can catalyze or they can function most. And we say these are mild reaction conditions that enzymes need to catalyze their various biochemical reactions. Now let's look at chemical composition of enzymes. So what are enzymes made of? So enzymes, as you said, they are proteins. Most enzymes are proteins, but few enzymes are made of nucleic acids. So the enzymes made of nucleic acids are called ribozymes, but most of the enzymes we know are proteins. So they are conjugated proteins because they are always linked to different compounds to make them active. Now when we take every enzyme in action, it always has two parts. It has a part we call an apoenzyme, which is just the protein part, and a cofactor, which is the functional unit that binds the apoenzyme to make it active. So when we say a holoenzyme, the holoenzyme is the active enzyme, which is made of the apoenzyme, the protein part, and the cofactor. So the cofactor binds the apoenzyme to make it active. This means that without the cofactor, the enzyme or the apoenzyme can't be active. Think of this. The apoenzyme is the inactive enzyme. So for it to be activated, it needs a cofactor. And the cofactor must bind the apoenzyme to make it or to convert it to an active form, which is called a holoenzyme. So when we say holoenzyme, the holoenzyme is a conjugate of the apoenzyme and the cofactor. So the cofactor can be a unit, being an organic molecule, or a metal ion that binds an enzyme to convert it from inactive state to an active state. Now the cofactor, normally we can have types of that. We can have a metal ion, which is just a mere metal that binds the enzyme and make it active. We can have what we call a coenzyme. The coenzymes are just organic molecules that binds the enzyme. So you can have NADH, NADPH, these ones are not in the slide, and also FMN. All these are coenzymes. They are normally synthesized from vitamins and they bind loosely to the enzymes to make it active. We can have other cofactors which we call the prosthetic groups. Some enzymes have their cofactors bound permanently to it. So if the cofactor is bound permanently, or that is irreversible or tightly bound to the enzyme, then we call that cofactor a prosthetic group. So the prosthetic group cannot be reversed, but they are permanently bound to the enzyme, so they can make it functional. Now these enzymes with the prosthetic groups are always functional, or they can be controlled in the body by whatever an allosterism. So let's look at this pictures here. So if this is an enzyme, this enzyme must bind its coenzyme here so it can be active, so that you can bind the substrate to it. So we are going to look at how this mechanism works in the next few slides. Okay, so let's look at the roles of cofactors as shown here. Normally enzymes have a site that substrate binds, which is called the active site. Now for the active site to bind the substrate, the site must be in a good conformation, or it must have a good shape as the substrate. Now if the substrate is not in a good shape, cofactors bind the enzymes and cause conformational changes in the active site so that it can suit the structure of the substrate. So if you look at this, if this cofactor binds the enzyme, the active site opens and stretches enough so the substrate can bind it. The same way, if we have this enzyme active site being open with just a small hole here, here is where the cofactor must bind so that it can give the active site a good conformation for the substrate to bind. So the active site is given a good conformation, but so that the enzyme can bind by the binding of the cofactor to the active site or to any point on the enzyme. So cofactors, we say, they activate the enzyme because when they bind the enzyme, they render a good active site conformation so the substrate can bind. That is how cofactors play a role in an enzymatic reaction. Now let's look at how enzymes are synthesized. Now we can classify it. You can classify enzymes by their composition. So by their composition, you can have enzymes as monomeric. If the enzyme is just made of one protein, we call it a monomeric enzyme. If it is just made of few proteins, let's say 1, 2, 3, then we call it an oligomeric enzyme. But if the enzyme is made of several subunits, that is several protein units, then we call it a multimeric enzyme. Also, a multimeric enzyme or a multi-enzyme is an example of that is fatty acid synthase. The enzyme which synthesizes fatty acid in the body is made of several subunits. Another enzyme is called a polyketide synthase that have several subunits for synthesis of polyketides in the body. But these ones, because they have several actives or several enzymes, they tend to have several active sites. So it's just one complex protein made of several enzymatic subunits that catalyze series of reactions on the same protein enzyme. Now let's look at how enzymes are named or the nomenclature of enzyme. Now, so as the name implies, enzymes are normally named by the reaction they catalyze or sometimes by the substrate they catalyze. So to just get a name of an enzyme from a substrate, as seen here, the suffix ase is always added to the name of a substrate. For instance, if you have a lactase, this lactase is an enzyme that breaks down lactose. If you have a maltase is an enzyme that breaks down maltose. You can have a proteases as an enzyme which breaks down proteins. You can have peptidases as an enzyme which breaks down peptides. An example, you can have DNA polymerase that forms DNA polymers during transcription. So you can also have enzymes based on their systematic name. So enzymes can also be given systematic name, which they have 6 main classes, which we didn't talk about in this slide. But we are going to just mention them. We have 6 main classes of enzymes. So the 6 classes are all the enzymes in this world are classed under. We can have enzymes we call the oxidoreductase that catalyze an oxidation reduction reaction. We can have another one we call transferases that transfers one group from one molecule to the other. We can have another one we call hydrolysis, and hydrolysis that adds water molecule to break down a bond or to break down a big complex unit to a smaller one. We can have what we call lyases. The lyases cleaves a compound without the use of water. You can have ligases. The ligases, they tend to join 2 units together to form a complex one. Then you can equally get an isomerases. Isomerases isomerizes a particular reactor to transfer the one group from one position in the same compound to the other. So now let's look at how enzymes work. So as we said, enzymes work by just lowering the activation energy, as we said in the next or in the previous slide. Let's try to explain what an activation energy is. So if you take every reaction, we see all chemical reactions have an energy barrier, which we call an activation energy, that separates the reactants and the product. So the activation energy we say is the amount of energy needed to disrupt stable molecules so that they can take part or the reaction can take place. So let's take this reactant. This reactant compound would fuse 2 compounds or 2 elements. This compound has its own initial energy level. That is, every compound contains some amount of energy in it. Now basically, when I take every reaction, if the energy contained in the reactant is higher than the energy contained in the product, that reaction is called a spontaneous reaction. A spontaneous reaction. For every reaction, if the energy contained in the reactant is more than that contained in the product, it's called a spontaneous reaction. So if I take this one, if this is an energy profile diagram, I will say the energy here is higher than the energy here, which is that of the reactant, the product, sorry. So the reactant energy is more than the product energy. It is called a spontaneous reaction. Now when I take a difference in energy, since this one is higher than this, the difference in energy is always the product minus the reactant. So the difference in energy is going to give me a negative value. That difference in energy here is called Gibbs free energy. When we say Gibbs free energy represented by G, we say it is an amount of energy in a system which is needed to do work. So if that difference in energy, which is given by change in delta G, is a negative value, it means that reaction is a spontaneous reaction. So the difference between this and that, the reactant and the product, if it gives me a negative value, then we say that reaction is a spontaneous reaction. Inversely, if the product energy is higher than the reactant energy, so a curve which is likely to go like this, so which will be like this. So it's not nice. So if the curve goes like this, so with this case, this is the product and this is the reactant. So here the reactant and this is the product. So here the product energy is going to be higher than the reactant energy. Hence the difference is going to give me a product, sorry, a positive value. So the difference in the reactant and the product energy is what we call the change in delta G. So the delta G is the energy, or the G here is the Gibbs free energy contained in each of the compounds. If I take the whole of this system, the delta G is the energy, or the internal energy in the system that is needed to do work. And that internal energy can be contained in the reactant. It can be contained in the product too. So if the reactant on is higher than the product, it's going to give you a negative value for delta G, and that's why you say a spontaneous reaction. If the reactant energy is less than, as seen here, if it is less than the product, the difference, which is always the product minus the reactant, is going to give me a positive value. That positive value, we say, means it is a non-spontaneous reaction. A non-spontaneous reaction is the reaction that can't occur by itself unless you add an external source of energy. A typical non-spontaneous reaction is photosynthesis, that is conversion of carbon dioxide and water to glucose and oxygen. That reaction is a non-spontaneous reaction that needs an external energy, which is sunlight, before it can occur. So if the reactant energy is less than the product energy, then we say that reaction is a non-spontaneous reaction. If the reactant energy and the product energy are the same, if they are the same level, then we say it is an equilibrium reaction. Equilibrium reaction. Bear in mind, enzymes act only on a spontaneous reaction. So enzymes act only on spontaneous reaction to convert them to product. So if this, given this profile diagram, this reaction is a spontaneous reaction, where the difference is energy is going to give me a negative delta G. Now when I take every reaction, there is a minimum amount of energy which I must overcome before I get to the product. So look at this. If I want to convert these two compounds, that is AC elements, if I want to break this one to the separate ones, I have to add in an energy. I have to attain an energy like that before I can break it down. So the minimum amount of energy required to break this bond to form the product, or the minimum amount of energy which is needed to just disrupt this stable compound, is what we call the activation energy. So if I take this as an energy diagram, this is the energy of the reactant, and here is the highest energy which can be attained by the reactant. Now the highest energy state of the reactant is called a transition state. Transition state. And the molecules there, when they are become very weak, when the bond is in here, the transition state become very weak. The molecules there are called activated complex. Activated complex. So if I have this molecule, let's say this profile diagram, the difference in energy between the ground state or initial energy level of the reactant and the transition state is what we call the activation energy. Activation energy. Now, if I add an enzyme, which is the red one, this enzyme is going to bring down the activation energy from the top here to the down here. For that matter, I'm just going to need a less time to get to this energy level before I can get to the product. So when we add an enzyme, we see enzymes increase the rate of a reaction by just lowering the activation energy. So the activation energy means that the energy required to break the bond, that is the minimum energy, is going to be lowered. So if the minimum energy is lowered, then the reaction can occur very fast within the shortest possible time. That is how basically enzymes catalyze the rate or increase the rate of a chemical reaction. They just lower the activation energy so that the reaction can proceed very fast. The reaction can proceed very fast. Think of this, how enzymes lower. Let's say this man wants to roll this board over this hill. This hill, we say, is the activation energy difference, which must be covered by this man before you can roll this ball over this hill. Now, if this is without an enzyme, this difference from where the man is standing to the highest energy level is what we call the activation energy without the enzyme. Now, if I have this one, the difference between the product energy and the reaction energy is called the net energy released from the splitting of the lactose or the splitting of the bond. So this energy difference is what we call the Gibbs free energy. That when you get a negative value, it means it is spontaneous, but when you get a positive value, it is not spontaneous. So this reaction occurs very slow, and it will take a longer time for this man to roll this ball over this hill. But if you add an enzyme to it, as seen here, that is here just an illustration of trying to break a lactose without an enzyme. It can happen, but it will take a long time, just like you trying to roll a ball over a hill. So if you add an enzyme to it, which is a lactase, it's just going to break the bond very fast. And the reaction, though, how it's catalyzed, just lower the activation energy. So here, the enzyme will just bring down this hill from the longer distance or from the longer difference to here. So this man will not need much energy to roll the bond over the hill. So the enzyme just decrease the energy required to reach the highest energy. So with this one, the reaction is going to proceed very, very, very quick. So now, what is the difference between an enzyme and a protein? So as said earlier, enzymes are proteins, and they are all derived through the central dogma, that is the protein synthesis. Protein synthesis. All enzymes are protein with the exception of few enzymes that are RNAs, and those are called ribozymes. So not all proteins are enzymes, but all enzymes are protein, with the exception of some few enzymes which we call ribozyme. So now let's discuss the active site of an enzyme. So when an enzyme is to catalyze a reaction, the substrate always binds the active site. So the active site is a region on the enzyme where the substrate can bind. The active site has two parts. We have the substrate binding site where the substrate can bind. Then we have the catalytic site where the substrate is broken down to form the product. Now the active site of the enzyme is the place where the substrate binds and at which the substrate occurs or the catalysis occurs. Now when the enzyme binds the substrate, they form what we call an enzyme substrate complex. Before this complex is broken down from the products and the free enzyme. When the enzyme binds the substrate, they form an enzyme substrate complex. Then this complex is now broken down to form the product and the enzyme. So the active site, you say, is just a region on the enzyme where the substrate can bind and gets transformed, or let's say catalyzed to the product. So this is how it works. If this is an enzyme, the substrate binds the active sites. The active site is always complementary to the substrate. Then this substrate is being converted to the product, as in here, broken down. So here this is what we call an enzyme substrate complex. The next step here is always the enzyme product complex. Before the product is released, then the enzyme is also released, regenerated. So the enzyme, as we defined earlier, itself is not destroyed in the course of the reaction, but it just converts a substrate to a product. So substrate approaches the enzyme in an enzymatic reaction. Then the enzyme binds the substrate at the active site and forms the enzyme substrate complex. Then the substrate is transformed into the product. The product is released and the enzyme is recycled. That is how enzymes catalyze the rate of a reaction. So an enzyme activity is just an ability of an enzyme to transfer or to transform a particular substrate through a product. So the enzymes we say are never expressed in terms of their concentration, but they are expressed in their activity. So when we say an enzyme activity, then we make reference to the most of the substrate that can be converted to a product per unit time. So if you say an enzyme has a higher activity, it means the enzyme can convert most of the substrates to um, a product within the shortest possible time. The rate of a chemical reaction is the rate of the appearance of the product, or the rate of the disappearance of the substrate defines the enzyme's activity. So if you say an enzyme is having higher activity, it means the rate of the appearance of the product, or the rate of the disappearance of the substrate is very fast. It's very fast. So normally we test the enzyme activity using what we call a spectrophotometer, which takes the absorber, the concentration of the product formed in a particular reaction. So let's look at factors that affect an enzyme activity. Then we are going to look at graphs that illustrate these factors. So how enzymes affect how quick a reaction can go, the reaction rate. These factors affect how quick the rate of a reaction can proceed. You can have substrate concentration, you can have enzyme concentration, you can have temperature, you have pH, you can have activators, and you can equally have inhibitors. So let's have just a pictorial view of how these factors affect the rate of a reaction. So let's look at how temperature affects, or the graph of how temperature affects the rate of a reaction. So this is just a graph that illustrates how rate of a reaction depends on temperature. So as I said, most enzyme works best at where we call an ambient temperature. That is always between 30 and 40. So here, this graph means that as you increase the temperature of a reaction, the rate of the reaction increase like that. To increase as you increase the temperature. So you get to a highest point we call an optimum temperature. Now this temperature is where each enzyme has its own best activity. So at this temperature, each enzyme is said to have its best activity. So around, let's say, 30 to 40 degrees, most enzymes are optimum. It means they have a highest enzymatic activity. Now as you proceed upward, if you try to still increase the temperature, then the graph will drop just like that. So it will just drop, meaning that the enzyme activity is going to drop. So if you increase the temperature above the optimum temperature, the enzyme is going to be denatured. So the denaturation is what is going to cause the decrease in the rate of a reaction, or the decrease in the enzymatic activity. So let's look at this. As the temperature rises, now the molecules have more and more kinetic energy, and this increases the chance of successful collisions, and so the rate increases. Now, the certain temperature at which the enzyme catalytic activity is greatest is what we call the optimum temperature, which is around 37 degrees Celsius. Now, if you go above this temperature, then there's going to be a denaturation of the enzyme. So when we say the normal body temperature of a human being is 37 degrees, it is so because this is the temperature where most enzymes have their optimum activity. So this temperature of a human system is that of the optimum temperature of all the enzymes in the human system. Now let's look at how also pH affects the rate of a reaction. So this also means that as you increase the pH like this, enzymes are proteins and they are made of amino acids. They have carboxylic acid functional groups, they have amino acid functional groups. So temperature or pH tends to change the charges on the amino acids. So if you tend to have a highly acidic pH, then you are going to confer a positive charge on the enzyme. If you tend to have a highly alkaline pH or basic pH, you are going to confer a negative charge on the enzyme. But if you confer an optimum pH of around 7, then the enzyme will have both charges. Now, if you have a highly acidic pH, because of the conferred positive charge, the negative charge becomes non-functional, so the enzyme's activity is very low. As you increase the pH to the optimum, or let's say the neutral one around 7, most enzymes attain their best catalytic activity. So with the pH where most enzymes have their highest activity is called the optimum pH or the optimal pH. If you proceed further to an alkaline, it will drop. So the difference between the pH and the temperature, as in how they affect the reaction rate, is that for the pH, as you get to the optimal, the reduction is very slow and gradual. But for temperature, as we get to the optimal, the reduction is going to be very sharp and quick. That is, there's going to be a sharp denaturation of um, the enzyme or the protein. Now let's look at these two, how substrates and enzyme affect the rate of a reaction. Now this is the dependency of the rate of a reaction on an enzyme concentration at fixed substrate concentration. So let's say we have enough substrates, then we increase the rate. Now the enzyme concentration, as we add more enzymes, we tend to bring in more active sites. As you add more enzymes, we tend to bring more active sites. So if you tend to move more, bring in more active sites or more enzymes, then the rate is going to just increase linearly. So as you increase the enzyme concentration, the rate of the reaction also increase linearly like that. So the dependency of the rate of a reaction on the enzyme concentration is that when you increase the enzyme concentration, we are going to gradually increase or linearly increase the rate of a reaction. At the same time, if you have a substrate, how substrate depends on the rate of a reaction at a fixed enzyme concentration. If you increase the substrate concentration, here you have a fixed concentration of enzyme. If you increase the substrate concentration gradually, the rate of the reaction will increase at the beginning, just like you see here. So here the active site is not full, the enzyme is there, but the active site is not full. But as you increase the substrate concentration, the active site is going to be full, and that is what you call, or you say, the enzyme is saturated. So the saturation point is a point where the active site of the enzyme is occupied. Now, when the active site of the enzyme is occupied with enough substrate, then there's going to be what? No increase in the rate. That is, there's going to conversion of no enzyme or no substrate to a product. Hence, even if you keep increasing, the rate of the reaction will remain constant. So that's how enzymes depend on, or the substrates affect the rate of a reaction. Now let's look at last thing we call an inhibition and inhibition. So enzymes inhibition. So instead of enzyme inhibition, it is just how enzymes can be inhibited. So some compounds compete with the substrate for the same active site. Let me just talk a little about this. Some compounds compete with the substrate for the same active site. So if the substrate and the compound have the same structure, that compound, which you call an inhibitor, can bind the substrate, can bind the active site of the enzyme, and blocks the substrate from binding. Now, if any chemical have the same structure of the substrate and can bind the enzyme and prevent the substrate from binding, that compound is called an inhibitor. So inhibition is just involved in the blocking of the active site of the substrate by different compounds. We have two main types of inhibition. We have reversible inhibition, where the binding can be reversed, and irreversible one, where the binding can be reversed. Now the reversible one, you can have several types. You can have competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. All those ones are types of inhibition that inhibitor can have on an enzyme. Now, if the substrate and the inhibitor have the same conformation, then the inhibitor is going to compete with the substrate for the same active site of the enzyme. So with a competitive one, the substrate and the inhibitor have the same structure. So the substrate can bind the active site, then the inhibitor can also bind the active site. So the inhibitor, we say, can compete for the same active site of the enzyme. This is what we call a competitive inhibition. Then with the non-competitive inhibition, we also have a site on the enzyme called an allosteric site. So if that site is bound by an inhibitor, that site, the binding of the inhibitor will change the conformation of the active site, so it can bind. So with the non-competitive inhibition, the active site of the enzyme will change when the inhibitor binds the allosteric site. And that will prevent the substrate from binding. Then with the uncompetitive, the active site, the enzyme or the inhibitor can only bind the active site or can bind different places of the enzyme when the substrate binds. So with the uncompetitive, the enzyme and the substrate will bind first, then the inhibitor will bind and make the enzyme substrate complex non-functional. And that's not in this slide, I just want to explain it so you can add it to what you have. Now let's look at how enzymes are used in clinical diagnosis. So in the hospitals, enzymes are used to discover diseases. Enzymes are basically found in um, various organs in the cells or in the body, like kidney. It can be in the liver or even the muscle. So let's say if an enzyme used to be in the liver but it is found in the blood serum, then we can see, or let's say the urine, then we can see there's a damage to that organ. If an enzyme is confined only to the liver and we test for that enzyme in the blood or the urine, then we realize that that enzyme occurs at high concentration in the urine or the blood. You can see there is a damage to these organs. So they are used to assess how well a particular body system or how well a particular organ in the body is working. Or has there been any tissue damage? So if there's a damage to the liver tissues, then the liver cells is going to expose some enzymes in the liver, in the blood, or even the urine. So common enzymes that are used for clinical diagnosis are these ones we call an alanine aminotransferase. You can have glutamate pyruvate transaminase. You can have an alkaline phosphatase, the most important enzyme. You can have an amylase. You can have aspartate aminotransferase. You can have creatine kinase and lactate dehydrogenase. All these enzymes are confined to the various tissues. Like for instance, the alkaline phosphatase is confined to the liver. So if this enzyme happens to be occur at higher concentration in the muscle, or let's say in the blood or even the urine, then we can possibly see there's a damage to the liver cells. So that's the basic on enzyme. Don't forget to like, subscribe, and also share to who is in the comments after watching, so you can receive every bit of update from this channel. This is Nox Captain, trusted process. Thank you.